graduate. She graduated last year, just a year ago, and I had the privilege to be on her committee. She did a really amazing job. So when we were thinking of people to invite, she automatically popped into my head. And so Madison is going to talk about how we can use paleolimnology to inform archaeology and other sciences with new developments, new and exciting developments that she has contributed to. So I'll just leave the stage to Madison, and she can present her what she's prepared for us. And afterwards, there will be a question period. So don't hesitate to stay, and we can have a chat.
All right, sounds good. Thank you for the introduction. All right, so let's get started here. So today, this talk I'm going to be doing, it kind of reflects a review that I worked on during my PhD. And what it really is meant to do is kind of like look at the state of the field and kind of think about ways that we could move forward and integrating paleolimnology with, archaeological research. So this is the review that I will be going on over briefly. If you are interested in more details, you'll be able to find them in this review. Today is going to be a high level kind of overview of it. I also should say that the views in this presentation do not represent the work I'm currently working on at Statistics Canada. So what is the objective? What I wanted to do was provide a review of proxies, specifically molecular biomarkers that can be found in sediment or paleosols, and then kind of incorporate them into a framework that illustrates how researchers like us can select these multitudinous proxies and biomarkers and so many things that we can detect, but how we select them to build better and stronger study designs. So the rationale here is, the first part is I provide a systemic review intended for the paleolimnologist audience, so you guys, and kind of look at the state of the art of the literature as it currently is for paleolimnological reconstructions of archaeological sites. And then kind of go over and try to standardize definitions for key proxy characteristics. And we'll talk about more about these in detail later. And then incorporate these standardized definitions into a framework that allows researchers to choose multiple proxies. And then there's some tidbits at the end of new future directions that I can demonstrate that will be most likely very interesting to researchers. So the overview of the talk will be a summary of the review, discussion on the framework, and how that framework can be integrated into study design and the utilization of decision matrices, and then those tidbits at the end for future discussion. So what did I find in this review? Well, first off, let's kind of set the settings for this review. Specifically, my thesis at the time was related to chemical proxies, specifically biomarkers. But I was kind of interested in how, archaeology and paleontology intersect, and how proxies can be used to kind of build a story that complements archaeological research. In order to do this, I started hunting for papers that related to chemical and biological proxies. So these guys here. So just to kind of reiterate, this is a classification system suggested by USGS for categorizing proxies. But biological basically means anything that comes from animal remains. So like diatoms, charcoals, the remains of plants, pollen, spores, and insect assemblages. Whereas chemical is more the single atom, if you're thinking of metals or isotopes or complex organic compounds, if you're thinking of biomarkers. So those are the groups of proxies that can be found in sediment or paleosol that can be used to build a story of archaeological sites. So how did I do this review? I tried to lean on systematic reviews used in epidemiology and other biomedical sciences. but it was fairly difficult here, and that's something that we're kind of going to work on and discuss. But I searched through multiple databases, including Scopus and Google Scholar. This was how I built the Boolean search term that you type into the search bar. So you have, basically this would be in brackets, and then this human limited search term would be linked to this matrix limited search term with an and you can build a complex Boolean search term that would find paleo or archaeological related papers from the soil, sediment, or peat, and that use proxies or biomarkers. However, because of the inconsistency in terminology in the literature, this was a fairly challenging way to do it. So additional searches were performed by, I would find a few papers and I would go look through their citations, see who they would cite, and then track down more citations that way. Studies were excluded if they were not published in English. There was a time limit, so if they were for reconstructing post-industrial age, so after 1760, the anthropogenic impact. So I wanted to look at more of the archaeological pre-industrial age reconstructions, and reviews were excluded as well. As I mentioned before, only biological and chemical proxies were included in this review. And just for simplicity, I limited the matrices to sediment, peat, and soil, though ice, and there are other types of paleoarchives as well. But these were the ones that were my main focus for this review. And from each study, I would try to extract the proxies used, the culture investigated, the location of the sampling, and the sample matrix. In total, about 89 studies qualified to be included in this review. And this here is just a subset of the papers that I would extract. So you'd have the reference here. And this is an example of the metadata that I would collect out of each one, including, the country where the sampling occurred, the culture that was under investigation, what was the oldest occupied period detected and from the samples, whether it was paleosol or sediment cores or peat cores that were used, how many proxies were used, what kind of proxies they were, so sterols, bile acids, or pollen. Were multiple proxies used? And I'll discuss orthogonality later. So this is just from the review. Of the 89 studies, I only included and went into depth on proxies that had been used by those studies. And what I found was for biological, charcoal, diatoms, arthropod assemblages, and fungi spores were the most commonly used from these 89 papers. And for the chemical biomarkers, we had a wider range. We had isotopes, metals, we had PAHs, monosaccharide anhydrates, and the list continues. It's fairly extensive, which is interesting. And this is an interesting synthesis that I was able to do using ArcGIS. So because I had where all of the studies were performed, I could go back and using a map here, I can say that, archaeological reconstructions via paleolimnology or, using paleosols, you can see how many studies have been performed in different areas. So the darker the color, the more study sites there have been. And you can really see that it's very localized to very few specific areas where there have been many studies. And the lighter areas here indicate areas where there just haven't been that many, paleolimnological studies. And for us as researchers, this represents something pretty good in the sense that, there's still lots of room for us to explore and expand our investigations. So from this review, just as a high-level summary here, I have a number of observations that were kind of just summarized it. So 56% of the 89 studies were published since 2015. And this indicates a rapidly growing field, which is always good. The studies were conducted worldwide, but research activity is uneven, and there are many under-investigated areas. So this presents numerous opportunities for future researchers to expand into these areas. And however, the absence of consistent terminology made typical search methods difficult, and ultimately this complicates, data integration and regional syntheses that can be performed, and to really strengthen the body of evidence that we have that paleolimnology can support archaeological investigations. And this is a really, this lack of coherence is a missed opportunity for the field because it complicates the integration of archaeological data with other broader climatological and environmental data from paleontology. And I think in the future, what a lot of people will like to see is this integration between these three areas to build a really comprehensive picture of what was happening back then. And finally, multi-proxy studies are becoming more common, but they are resource demanding and difficult to design. So this last observation is really what drove us to identify a framework that can be applied to improve the study design of multi-proxy studies using paleolimnological methods. So what is this framework that I keep talking about? So it's basically based on these, and I should emphasize that I have not really invented any of these terms. These terms have been lingering around in the literature since, paleolimnology started. However, there's been a few attempts to kind of really define them, but they haven't really been brought into like a standardized state within the field. And I think this can really benefit the field in making not only the literature easier to understand, but easier to integrate and potentially to choose your proxies as well, which is the angle I'm going to be discussing here. So this flow just indicates a general path of a proxy through its initial release due to some induced historical environmental change. So increased heat, temperature from climate induces more pollen release, and that pollen goes off and is deposed somewhere in the environment, hopefully rolling into our lake sediments. And then years later, we come along and we try to detect this from our lake sediment cores and hopefully derive some environmental inference from it. This here is just if I have a similar proxy. So again, I'll use the example from pollen from another lake. How well does it correlate between the two? And if I bring in a second proxy that isn't pollen, let's say it's like terpenoids or something, or other archives, how do these relate and connect to each other? So that's what the diagram basically shows. And then each of these terms here tie into these different areas or times of the proxy life. So specificity, what do I mean by specificity? So specificity describes the particular source or sources of a proxy. So a proxy is known to be highly specific if it comes from a very narrow source identification. So something like bile acids, which are typically from the fecal input of animals. It's very narrow where it comes from. It can be used to tightly link to some source. But you have other variables like bulk measurements of biogenic silica or total organic carbon that have multiple sources, and these are proxies of low specificity. The next here is something else that's relevant to proxies, is something called dispersibility. So how is your proxy dispersed through the environment? Is it aeolian transported? Is it released into the wind and kind of blanket deposited over a wide area? Or is it a more narrow deposition, like large macro scale pieces of charcoal that kind of go into your lake and sit there and maybe span multiple intervals? So this is something to consider when choosing your proxy. Resistance to degradation. So what happens to your proxy once it's been sitting in the sediment? So things like molecular biomarkers are susceptible to diagenesis, which results in structural changes in the organic molecules that occur after deposition, usually because of microbial activity, some to abiotic changes induced by the environment that they're sitting in. But even biological proxies like pollen or arthropod assemblages can be affected by dissolution and changes over time while sitting in the sediments. So how resistant your proxy to degradation indicates how reliable your proxy is overall. Some proxies are mobile in the sediment. So this means that they've been deposited. This is all well and good, but certain metals, for instance, if we're in a water-bound sediment, can actually migrate up or down a core just based on concentration diffusion. Whereas others like hydrophobic biomarkers may be more likely to absorb onto specific particulates and stay within a small band. So something like mobility can affect your profiles from your sediment cores, because even if they were deposited in a narrow band, they may not stay in that narrow band in the core. So detectability here is, this kind of largely pertains to what happens in the lab. How do you get these molecules out? So some proxies, for example, like polyaromatic hydrocarbons and sedimentary DNA, you know, they're hard to get out of the sediment. There's multiple cleaning steps, and there's a lot of care that goes into the sampling. Whereas there are other methods that are, you know, potentially use less harmful, like green or more better green chemistry techniques, or they can be analyzed without extraction. So this kind of this ties into how the proxies will be chosen later. Reproducibility has to do with if you have something like a highly reproducible proxy will be pollen. So you can say that from this lake I've sampled this, and this lake I've sampled pollen as well. Do those match up? I mean, there's some changes in dispersibility and also lake type that will affect this, but overall, is that proxy reliable, and is it able to repeatedly tell you about a certain historical environmental change across different sites? The last two here are accuracy and sensitivity. These are probably the most complex, but accuracy has to do with how accurate the proxies are able to track the environmental changes. So basically what this ties into is that you should be able to validate your proxy from other environmental archives. So something like Landsat imagery to confirm, you know, lake history, and see how well your proxy correlates with that external other archive and integrate that data. Sensitivity is highly variable. So it's how sensitive your proxy was to environmental changes. But this one is highly dependent on what your sampling profile is, whether it's every 0.5 centimeters or 1 centimeter. So that temporal resolution that you get from your core can really influence the operational sensitivity of your proxy. So considering all of these together, I also wanted to tie in the concept of orthogonality into the framework. So what do I mean by orthogonality? So you can see here, this is the broad groups of the biomarkers and other proxies that I found from the 89 studies. And I kind of broadly classified them into 3 broad groups: animal, environment, and fire. You can see there's some crossover here. So some crops are more indicative of the environment, whereas some are actually indicative of human farming activity. So this would be pollen from those crops. And the concept of orthogonality is to say, if you want to build a multi-proxy study that kind of that has the highest, the strongest evidence for inferring environmental changes caused by humans, you want to draw from each of these orthogonal groups because they come from different and independent stories. So they all provide information from the same site, but they complement each other in what they can tell you. So like the animal component will tell you about, you know, increased fecal input from either human sources or pastoralism, mining, whether that happened there. The environment will tell you if there's been any broad shifts in vegetation, largely from people clearing wide areas of land for agriculture, and then that presence of those crops. And fire for, you know, were they mining or were they doing slash and burn, you know, agricultural techniques to clear the land? So having a proxy from each of these groups really builds up your evidence in a multi-proxy study. So now I've kind of gone through the framework. How do you apply that framework into choosing studies or to choosing proxies for your study design? I mean, it is really challenging to ultimately choose proxies for archaeological investigations. There's several factors that go into deciding this, including cost, available instrumentation, available expertise, and what is the relevance of these proxies to the culture being studied. However, there is increased pressure to design these studies because they are becoming more common. So this is just a list of example references that they really embrace the orthogonal multi-proxy approach in their study designs. But this really only accounts for 15% of the 89 studies that I presented in the review here. However, it is obviously becoming more popular. So one was published in 2012, and then they've been increasing ever since. Okay, so it's really becoming an important consideration for publication to get these future studies and applying these multi-stream orthogonal evidence approaches with multiple proxies. So how do you go? Yes, so framework plus something called a decision matrix equals a study design tool. So what is a decision matrix? Some of you may be familiar with it, but it's typically a tool that is commonly used in engineering and business to decide which object or materials are optimal based on common criteria. And it's a qualitative technique. Typically, they're set up like this, where I've put in, these would be the proxies you would be considering along the top. And on the left here, you would have criteria. Based on these criteria, you're able to rank these proxies and then eventually sum the proxies for each proxy score, so the scaling here for the best to worst would be -2 would be, a poor performance of that proxy for that criteria, whereas a +2 here would be the best of all of the proxies in this list. And then if we want to factor in orthogonality here, we can choose which proxies we want to take. So let's say if we take proxy 1 and 3 here, we can sum these scores and then apply an orthogonality factor here. 1, if there is no orthogonality, so i.e. it all comes from a vegetative source. So it can only really inform you about vegetation in that archaeological site. 2, if 2 of the different orthogonality groups are included. So this could be like a fire paired with a human or animal indicator, and kind of linking the fact that if your fire instances increase with the amount of fecal input or some human animal indicator, they're more likely to be correlated because they're increasing at the same time, and you have more stronger evidence based in that study. And 3, if from proxies are selected from all 3 different groups, so vegetation, fire, and animal, this is the highest factor that you can apply, and you multiply that by the sum of your proxy scores. So the objective is to select a combination of scored proxies that has the highest study score while also taking into account practical considerations such as cost, logistics, and researcher capabilities. So how do you apply this? I'm going to walk you through kind of a scenario that we outlined in the paper. So we're researching culture X. They were semi-nomadic hunter-gatherers and hunted large marine mammals, i.e. whales. They had small localized hearth fires for cooking and warmth. And as a research facility, we have access to instrumentation to measure PAHs and sterols. But we also have 2 potential collaborators here. We have collaborator A that can analyze microscopic charcoal and plant terpenoids. And we have collaborator B that can analyze insect fossils and coprophilous fungi spores. So how do we pick the best studies design knowing these factors? Well, we can go through the ranking here. So I'm just going to use charcoal as an example. So you can see here, I've tied in all the terms from the framework that I applied earlier. And if we're going to go through each step here, so for charcoal, it was given 0 because charcoal is specific to fires, but is not specific to human fires specifically. It can come from natural fires as well. So it was given a rank of 0. For dispersibility, charcoal can be deposited locally, which is important for a study of a culture with only small localized fires. Resistance to degradation based on past studies. Charcoal has been very resistant to diagenesis, and its preservation is facilitated by cold conditions, which is very helpful. So a good one there. Mobility, based on past studies for this, charcoal is not mobile in the sediments, which is great. Detectability. So this is kind of an aspect where you can tie in ease of analysis. So for instance, charcoal is extracted using hydrofluoric acid in some cases, and charcoal particles can be broken down during this process, resulting in artificially high charcoal numbers. And also you can, add in the fact that hydrofluoric acid is very toxic and hard to work with. Those are both negatives, so we rank it as a -1. Reproducibility, again, past literature has shown that charcoal is pretty much fairly reproducibility. It can have some issues with deposition. And also analytical method may influence the charcoal numbers. So we're going to give it a nice neutral 0 there. Overall, though, charcoal has been shown to be a reliable indicator for fires. That has an accuracy, a score of 1. And sensitivity. So larger charcoal pieces can reduce sensitivity, but because we're using microscopic charcoal and not macroscopic charcoal, our resolution will depend on the intervals analyzed. So whether we take a 0.5 centimeters or 1 centimeter intervals. And so it depends more on the sampling technique than it does on the ability of charcoal to be sensitive to the occurrence of fire or past fires. So we ranked it as a 1. So then we have the sum. We basically go through the process of this for everyone, considering the culture that we're analyzing. And we can generate that proxy score for each of these. So if we were in that perfect world where we can have both collaborators and look at all of these proxies together, the highest possible score is 31, since all proxies are present from the orthogonal groups, whereas we have fire indicators, which are charcoal and PAHs, vegetation indicators, plant terpenoids, insect fossils and sterols, and human and animal indicators, including cannabinol, sterols, insect fossils, and fungi spores. And since we have, they're all present from all the orthogonal groups, they can be multiplied by 3. So the study score with all of the proxies here would be 93. But it's not the perfect world, so we can only use some of these proxies. But you can use this decision matrix to kind of weigh in and decide which ones will be the most useful. So looking at this, I can see that PAHs have the lowest score. And mostly this was because culture X had only small localized Hirsch. They didn't do big slash and burns. They probably did not produce enough PAHs to track a decent fire record. So PAHs were removed from the list. Then we looked at collaborator A. So again, we're doing the analysis on the sterols, but they can bring in the analysis from the plant terpenoids or the charcoal. And based on that analysis, and they have proxies from the 3 different orthogonal groups, they have a combined study score of 51. Where if you go to collaborator B, who analyzes the insect assemblages and the fungi spores, they only have a study score of 40. So the best ideal study design would include collaborator A in this to have the better study score. So what does all this mean in terms of designing an approach for study designs? Is that we could really stand to standardize terminology to make things like systematic reviews easier. And if we have a consistent framework and consider orthogonality, we have a better approach to designing multi-proxy studies. And once we have this framework, it's easier to integrate and then synthesize these studies together to have a more systemic, holistic approach when approaching, when building, incorporating your paleolimnologist study into the body of archaeological work, especially when an archaeologist wants to come in, bring in their data, and then you have a climatologist wants to come in and bring in their data, and then you have our paleolimnology expertise, and we want to bring in ours. So the ability to integrate all of this depends on having that data available and in a coherent, consistent manner that it can be applied to a variety of different synthesis studies. So something also kind of a neat tidbit to consider going forward in the future is that if we do work on, bringing our data into the cyber world, there's the possibility for analyses like this. So this is a map that can be used to find areas where there should be a lot of sites for paleolimnological investigations. So all I did here was I found an online database of archaeological sites and also representative of human density over time for the last few thousand years. And then I took a map of lakes and combined it into a density map for lakes. So areas where there are a lot of lakes and ideally good sites for this would have been highlighted. And then you can correlate that population density map, that historical population density map, with that lake density map to see where there would be good sites for archaeological investigations. So that's what this map represents, where if it's a darker color, these would be ideal sites for archaeological investigations using paleolimnology. And as you can see, there's a lot of future opportunities here for us, which is excellent. Another thing, and this was explored in my thesis a little bit more, is the discovery of new biomarkers. Maybe we want to have some easier to detect biomarkers, or if we want to flesh out the story some more. I have articulated a method that can be used to discover new proxies. It's called sediment omics. And basically it borrows from the metabolomics holistic approach of doing an untargeted chemical analysis. I'm not going to go into too much details here, but based on your sampling paradigm, you can discover new biomarkers by comparing two different sediment types and looking at the chemical composition of these to figure out, if this sample was from a boreal tree forest and this sample was from the tundra, what chemicals are present in the boreal that aren't present in the tundra, and vice versa. And maybe those can be used as future biomarkers. So lots of future things to investigate. And these are my acknowledgements. Thank you to my funding sources, my past committee members, my supervisor, mentors, and everyone that has helped me along the way. Thank you.
Thank you, Madison. It's really interesting. I find it, it's a really good approach to try and have a quantitative evaluation of your plan, your multi-proxy study. I wouldn't have thought of that.
I think the only reason I thought about it is since I've jumped around so many disciplines at this point, and it's just like, oh, okay.
It's really, really useful, and I think I'm going to use it for my next project.
Awesome.
So does anybody have any questions that they would like to ask Madison about her presentation? Just if you have a question, just ring in, just because I don't think I can see everyone.
I can ask a first one. Just, well, maybe two. One might be easy. Just from the beginning, you started looking only at paleolimnological study that use lipid biomarkers, right?
Yeah, mostly.
Okay.
Yeah, in the beginning, mostly because I built it off of my thesis, which was related to lipid biomarkers, but I really wanted to expand this to make it a method that is useful for people who are using whatever proxies are available to them.
And then if we think about the lipid biomarkers, I was also wondering if we should consider, your orthogonality, looking at proxies from different like fire, humans and things. If we should also be worried a bit about using, for instance, you might be able to use only lipid biomarkers for this kind of works, but then it's only like one analytical technique. And if you mess something up, maybe you might see a signal that is coming from, I don't know, an error during your analysis. Would it be useful to also, like in the orthogonality, think about having proxies from not only like one type, like the lipid biomarker, like also the charcoal, that you need another analysis to get to the result?
Yeah, no, that makes a lot of sense in the sense that, if you're taking the same lipid extract from the same sediment cores, and you messed up that extract, but you're still, doing the same analyses on it, you would expect bias. And oddly enough, the term orthogonality that I used is actually derived from the analytical sense that you've mentioning, because it was originally designed to talk about, if I have an ICP-MS detecting metals, but I have an AAMS or an AAS detecting metals, there are orthogonal approaches that detect metals concentrations, but different ways, right? So that was the original definition of orthogony that I kind of borrowed off of. But what I've presented here is kind of meant as a starting point. Again, like I'm just did my PhD thesis, so I'm very much a new person to the field, but it definitely needs some more development. But I think that the overall framework could be really useful going forward.
Thank you.
I have a question. I was very struck by something you wrote in a very early slide, which was the absence of consistent terminology was a problem.
Mm-hmm.
How did you deal with that?
Through a lot of slogging through citation lists and then going to the papers, clicking on the hyperlink and reading through it and saying, okay, is this good or is this not good? So, I mean, again, I'm borrowing heavily from a little bit of the epidemiological field where it is easy for them to have consistent terminology, right? Because, they're working on a very limited system where we're working on multiple systems. But I still feel like there is room for us to kind of improve that and have that consistent terminology so that we can really synthesize our bodies of work together to have these big climatological, archaeological papers and viewpoints.
I completely agree. I can't help feeling this. There's a lot of culture involved in the different sciences, and getting people to part from their favorite terminologies may be very challenging.
Yes.
So maybe we need a translator somewhere. I don't know.
Yeah, that would make a lot of sense. Thank you.
Yeah, and we can't impose terminology on anyone, I think, which is another difficulty. Do you think that the sensitivity of proxies is variable according to the region?
Yes.
For some of the proxies, so it would be difficult to assign a value to any one proxy globally. So
yes, I did try to kind of apply the framework in a more like localized setting. So it's kind of pertinent to the culture that's under study, right? Like when I was speaking about the small hearths versus like slash and burn agriculture, right? pHs would have been great for the slash and burn, but not so great for small localized hearth. And that I think that same kind of thought also applies to that specificity argument. Because like if you're using pollen in Africa versus using pollen in the high Arctic, yeah, you're more likely to have pollen preserved, maybe not the higher, but preserved in the Arctic than you are in Africa, where it's mostly insect-based pollination, and you might not have the big pollen dispersals that we get here. So yes, pollen in a general sense, like if you want to say global sense, can be specific, but in a local sense, not so much. And that's really where the culture that you're investigating comes into the decision matrix.
Okay. Ray Bradley has a question.
Thanks for the interesting talk, Madison. I have a question that you didn't mention, or a parameter you didn't mention, which is sedimentary DNA, as a proxy for, or an indicator of, for example, the presence of grazing animals in an environment which might have been introduced. And I mention this because we have a paper that will come out this week in Nature Communications, where the presence of sheep in an environment which were introduced into the Faroe Islands is indicated by sedimentary DNA. We have many of these other parameters that you mentioned too, we've looked at, but I think it was the arrival of sheep that was the most important indicator of a human presence in that environment.
So, I mean, I didn't use environmental DNA in the proxy, but I do talk about it in the review because there's some fascinating papers out of Greenland with Shearsham, who talk about different Arctic cultures and how, based on timing in the sediment core and eDNA compositions, they can say that, you know, the Thule were more prevalent here, or the Dorset were more prevalent, or the Sac. So there's definitely interesting work to be done there. But I think if you treat, still have to treat environmental DNA as one of the proxies in your decision matrix. And it can still be, that matrix can overall still be applied to that thinking. But yes, they can indicate orthogonal approaches, because you can have eDNA from vegetation, but you can also have eDNA from human and animal sources.
I agree. There's no one proxy that's going to give you all the answers. So, which is why we do multi-proxy work in the first place.
Yep, and also incorporating that orthogonality concept that Natalie was talking about earlier, where you have different detection techniques as well. So if your sampling was thrown off potentially of your environmental DNA in like one of the intervals, do you have something from a different detection method that can collaborate those results?
Yeah, I agree. Any other questions? No? Okay, well,